see the screen?
A division, start writing the attendance in the chat box. Only those who will write the attendance in the chat box will be marked present. Huh? If you forget, then you forget the attendance also.
Okay, can you see the screen? Am I audible?
Yes, ma'am.
Screen is visible?
Yes, ma'am.
Yes.
Yes.
Okay. Okay. Now let us start with the new chapter, that is enhancement of food production. Uh, this chapter, as you know, some of the part, whatever is deleted, that portion, deleted portion PDF has been sent to you. Once again, you can go through it. So whatever is there for the assessment, so that part we are taking. Okay. So enhancement of food production. In that, first part is biofortification. So what does it mean? Biofortification means it's a method in which crops are bred, that is produced for having higher levels of vitamins, minerals and fats, that is better nutritive value. And due to this, problem of malnutrition can be overcome. And so these are the objectives to be considered for this breeding program. So first is protein content and quality, oil content and its quality, vitamin content, micronutrient content and quality. So first, before we come to this point, let me tell you, biofortification is nothing but improvement of the nutritional value, or improving or increasing the level of nutrition in these crops, so that this will help to reduce the percentage of malnutrition, because every year, because of malnutrition, because of insufficient amount of food, or without nutritive value if the food is taken, so such type of food which is of no use, so many people die. So death, all because of malnutrition, can be reduced, that percentage can be reduced. And so that's why this is one of the important methods used in plant breeding. So biofortification is very important to improve the nutritional value of the plants. So what are these criteria considered for the improvement of the values? What are these nutritional values? Nutritional value of proteins, fats, vitamins, micronutrients, and along with that, not only quantity but its quality as well is also very, very important. So quantity and quality, both the things are considered for improving the nutritional value, or increasing the nutritional value. So biofortification can be achieved through conventional selective breeding practices, and also through rDNA technology. You know what are the conventional selective breeding practices means? For example, hybridization, the selection of the parents, where desirable characters, the parents considering the desirable characters are selected, and then their result is gained with the help of improved varieties. As well as, now last time in the last few chapters we have discussed the rDNA technology also, that means recombinant DNA technology, where we have discussed so many examples of plants where their different values, nutritive values are improved, and their production is also increased. As well as in case of animals also, nutrition as well as, that means quality as well as quantity, is increased by using the rDNA technology. So it focuses on making plant food more nutritive as plants grow or develop. Now what are the examples? Some of the examples are given, that is by using this method of biofortification, either conventional methods or by using rDNA technology. What are the examples? We'll see now. fortified maize having twice the amount of amino acids, lysine and tryptophan. So maize, last time we have studied corn. So by using rDNA technology, so this is one of the examples, fortified maize having twice the amount of amino acids like lysines and tryptophan. Then wheat also, Atlas 66 has a high protein content, and iron fortified rice has 5 times more iron. These two examples from the cereals, 2 3 maize, wheat, rice, these are some of the examples. In case of maize, amino acid content is increased. In case of wheat, its protein content is increased. Then in case of rice, its iron content is increased. So these are some of the examples. Then vegetable crops like carrot, spinach have more vitamin A and minerals. Then vitamin C enriched bitter gourd tomato have been developed by Indian Agricultural Research Institute. So these are some of the examples where in some cases amino acid content is increased, in some cases protein content is increased, iron is increased, micronutrients, minerals, vitamins, these are increased by using this method of biofortification. Okay. Now let us come to the next point, that is microbes in human welfare. There are different types of microorganisms which are useful for the human beings. So these all we will discuss step by step. Now you know that microbes are considered to be omnipresent. They are present everywhere. For example, they are present in soil, water, inside our body, other animals and plants also. These are some of the examples. Even they have a different range as per the temperature. That means they are present at high temperature as well as they are present in a very low temperature. Some of the examples, that is they are deep inside the gaseous, that is thermal vents, high as 140 degrees Celsius, deep in the soil, under the layers of snow, and they are also in highly acidic environment. So these are there is a high range of presence of microbes as per the environment. So this is just example. Now what are these different types of microbes? These are protozoa, bacteria, fungi, viruses, viroids, prions, etc. Now you know that all these examples, protozoa, different types of protozoa, some of them you know that they are disease causing, bacteria useful, harmful, viruses. So all these are some of the examples which are mentioned where these microbes are present. These microbes like bacteria and many fungi can be grown on nutrient media to form colonies that can be seen with the naked eyes. Now when if some of you have observed in the microbiology department or in some of the department, biotechnology or something like that, so nutritional artificially nutrient medium is used, and on which these microbes can be grown. So these are some of the colonies, just for your uh, just reference for to know, to show you these some of the examples I'm showing you here. These are the different microbial colonies. In this way these are produced, where it looks like the growth of cottony structure appearance, where here different colored microbes with the different colored colonies you can see. These are some of the examples. Okay, so these are the cultures, artificially produced one. Now microbes in the household purposes or the products, everybody knows while discussing the biotechnology, some of the examples we have learned also. Common example of production of curd and milk, you know everybody, where lactic acid bacteria are used. They are known as lab. Lab means what? There, that is a short form of lactobacillus or lactic acid bacteria. So microorganisms such as lactobacillus and others, commonly called lactic acid bacteria or the short form as LAB, grow in milk and convert it into curd. So these are the traditional, some of the examples which we have already discussed in the biotechnology chapter also. So during growth, the lab produce acids that coagulate and partially digest the milk proteins. So what are these? Small amount of curd and fresh milk that results into the product by inoculum or starter contains millions of lactic acid bacteria at a suitable temperature. So this, considering a specific temperature, the conversion of milk into curd or yogurt takes place in presence of lactic acid bacteria. And one more quality of this is it improves the nutritional quality by increasing vitamin B12. Then in our stomach also, that there these lactic acid bacteria, that means lab. I will be using the word lab for lactic acid bacteria now henceforth. Play a very beneficial role in checking disease causing microbes. This is also a very important thing. Now, probiotics, you also must be knowing, they are very friendly bacteria used for therapeutic purposes for the treatment, betterment of the human health. Now, the most commonly used microorganisms in case of bakery products, uh, milk industry, bakery industry, there are different types of microbes used, and traditionally they are being used for from so many years. You know that dough which is used for making bread is fermented using baker's yeast. So for MCQ, the question can be asked: dash is the microbe called as baker's yeast. So what is that? Saccharomyces cerevisiae. So this is what you should always remember. Number of traditional drinks and foods are also made by fermentation of the microbes. So these are some of the examples, different types of beverages. Then these food items, they are made with the help of traditional methods, traditional drinks and food products. They are used by using different microbes. Now this is also mentioned in your textbook also. That traditional drink of some parts of southern India is made by fermenting sap from plants. So some palm trees, sap of the palm is used and it is fermented, which gets converted into the traditional drink called as toddy in the southern in India. Then these are some of the examples where microbes are used to ferment fish, soybean and bamboo shoots to make food. So some of you might be using also fermented products made from fish, soybean, bamboo sticks or bamboo shoots, delicate bamboo shoots. These are. Cheese is one of the oldest food items in which microbes are used, and different varieties of cheese are known by their characteristic texture, flavor, and taste. The specificity coming from the microbes used. So, which type of microbe is used? That according to that use of microbes, the activity of microbes, the different types of flavors are made or different types of flavors are seen given to the products like these cheese. So these different types of cheese are known by the help of their variety of microbe or the type of microbe used in its production. Okay. And for example, the Swiss cheese is produced in such a way that large amount of CO2 is released, and that bacterium used, that microbe used is that Propionibacterium shamini. So this is the name of the bacterium for MCQ. These different names are already there in the textbook, you just underline that. So these are the different types of teas produced using different types of bacteria or microbes. Now this is one more example mentioned in your text also, that the Roquefort cheese is ripened by growing a specific fungi on it, which gives a particular flavor. So using different microbes, these different products are produced. Now in case of, now we studied in case of food products. Now in the industrial products, what is the role of microbes? Now there are different types of industries. These microbes are very much useful in the synthesis of number of products which are valuable to the human beings. What are these? Some of these are very, very important, like antibiotic production, then different types of beverages. These where the microbes play a great role. And you know that there are large fermenters, large vessels are there. They are called as fermenters for the production of these products. These large vessels, they are called as fermenters, where these microbes are grown. They are used on a large scale production industries. Now, fermented beverages, microbes, especially yeast, have been used for the production of beverages like wine, whiskey, brandy, and rum. Now, as for the type, how they are produced, they are known with the percentage of the alcohol and the, which way is used for its production, with distillation, without distillation, they are named by different names. And the same yeast, that is Saccharomyces cerevisiae, is used for bread making, commonly called as brewer's yeast. As I said just now, that this Saccharomyces cerevisiae is considered to be the brewer's yeast because that is used in bread making as well as in different formation of different beverages. So it is used for fermenting malted cereals and fruit juices to produce ethanol. So the production of ethanol using different methods and different microbes, especially this common microbe, they are used. And what is the raw material? Cereals, malted cereals, and the fruit juices. Depending on the type of raw material used for fermentation and the type of processing, just I said, that is with or without distillation, different types of alcoholic beverages are obtained. So what are these products which are used by distillation? They are whiskey, brandy, and rum, which are produced by distillation of the fermented broth. So according to the processing and the raw material used, the names are given. Antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease causing microbes. And we are familiar with the commonly used antibiotic, that is penicillin. And you know that the definition sometimes can be asked that what are the antibiotics? What are these antibiotics? These are nothing but the chemical substances which are produced by some microbes. They are produced from the microbes, but they kill or retard the growth of other microbes which are considered to be disease causing. So you should always remember this is the general information which is mentioned in your book also, that Alexander Fleming, while working on staphylococci bacteria, once observed a mole growing in one of his culture plates around which staphylococci could not grow. And he found out that it was due to chemical produced by the mole, and that the name which was given by him is penicillin. Penicillin was the first antibiotic discovered, and he named it as penicillin after the mold Penicillium notatum. So this is the product produced from Penicillium notatum. So the name was given as penicillin. So while working on the some of the culture medium, these staphylococci couldn't grow, couldn't show growth in one of his culture plates, and that was observed to be because of this, that is the Penicillium notatum mold, from which this penicillin was produced. So penicillin is considered to be the first antibiotic discovered. After penicillin, several other antibiotics were also purified from microbes, other microbes like streptomycin, gentamicin, neomycin, etc. These are some of the examples, and some of the examples for the treatment of different diseases. These are mentioned, specially greatly they are used, have been used for improving the deadly diseases such as plague, whooping cough, diphtheria, leprosy, etc. So different. So see how greatly these microbes are working, isn't it? Now, chemicals, enzyme, and other bioactive molecules. Microbes are also used for commercial and industrial production of certain chemicals like organic acids, alcohols and enzymes. Alcohols we discussed just now, that is the ethanol, its percentage, and as per the raw material and the processing and its percentage of the alcohol, the names are given. Now we will talk about the organic acids and enzymes. Okay, these are some of the examples. Now, which microbe is used, it is there in listed in your textbook also. You just have to remember these names, because for MCQ or for one answer sentence, this is very important that you should know the product and the microbe used. First example, that is Aspergillus niger is the microorganism, and it's that which type of microbe it is, fungus, and the product is citric acid. Then Acetobacter aceti, acetic acid is produced using Clostridium butylicum, butylicum, that is the bacterium, butyric acid is produced. Then Lactobacillus is the bacterium which gives lactic acid. So these are the products and these are the names of the microorganisms and these are the types of microbe. So you should always remember. Now here we can't help, we can just tell you all these tables which are given in your textbook. That is the name of the organic acid and the name of the bacterium or the microbe used, like enzymes and its microbe used. So like that you need to remember. So yeast, that is Saccharomyces cerevisiae, is used for the commercial production of ethanol. That is that. Lipases are used in detergent formulations and are helpful in removing oily stains in laundry. That is also mentioned. Microbes are also used for the production of enzymes. Now, lipases, these are used in detergent formulations which are helpful for removing oily stains. Remember, for MCQ also this question can be asked. Now, one more thing you should remember, that proteases, streptokinases, these products, they're used as clot busters for removing clots from the blood vessels of patients who have undergone myocardial infection leading to heart attack. Myocardium, you know that it is the heart, that is related to myocardial infection like heart attack, where the main reason is formation of clots in the blood vessels. So by using these proteases and streptokinases, these products, these clots can be removed. That means whatever the clots are produced in the blood vessels, they can be converted into normal blood. So these clots can be removed. So this is just example where these. Now another bioactive molecule, that is cyclosporin A, is used. How it is used? It is used as immunosuppressive agent. See, statins act as competitive inhibitor for the enzyme responsible for the synthesis of cholesterol. You know that the role of cholesterol. Some which type of cholesterol is useful, which is which is not useful or harmful to the body. Accordingly, these bioactive molecules are working, and statins produced by the yeast, that is Monascus purpureus A, have been commercialized as blood cholesterol lowering agent. Means using this yeast, using this microbe, that is the yeast, blood cholesterol lowering agent, it work as that means cholesterol level in the blood is maintained or it is lowered. So this is working very nicely in case of the generally in case of the patients where cholesterol level is increased, and because of that so many problems are observed, diseases are observed in the human beings. Now these vitamins, the next product of the microbes where they are playing the role in the production of vitamins, these are some of the examples. See here, you have to always remember the name of the vitamin and its microbial source, like that name of the enzyme and its microbial source. So vitamins are some organic nitrogenous compounds which are capable of performing many life sustaining functions inside the body. These compounds cannot be synthesized by humans except vitamin D, and therefore they have to be supplied in small amounts in the diet. As the human body cannot produce, cannot synthesize, they are supplied to the body in the form of food. So the diet should contain vitamins. But microbes are playing a great role. These microbes are capable of synthesizing vitamins, and hence they can be successfully used for the commercial production of many of the vitamins. For example, thiamine, riboflavin, folic acid. So all these vitamin B12, ascorbic acid, beta carotene, etc. These are some of the examples of vitamins which can be manufactured by using different methods like fermentation and using different microbes for the process of fermentation. So some of the examples which are mentioned in your textbook are B12, the microbial source is Neurospora gossypi, as well as Erythrothecium ashby. So these two names you always remember for the production of vitamin B2. These two are the microbial sources. Then vitamin B12, Pseudomonas denitrificans. Pseudomonas denitrificans is used in the production of vitamin B12. Vitamin C is produced by using Aspergillus niger. Now, these are some of the examples of the enzymes and their microbial source. Invertase by using Saccharomyces cerevisiae, pectinase by Sclerotinia, Libricina, then Aspergillus niger, lipase, Candida, Lipolytica, then cellulase, Trichoderma, Onigi. C. The MCQ type of questions can be asked. Vitamin C is produced by using dash dash microbe. Or the question can be asked, Candida lipolytica is used in the production of dash 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 vitamin or the enzyme. The name also can be given or like that. So vice versa, either the name of the vitamin or the enzyme or the product can be asked, or the microbial source can be asked. So MCQ, that means there's multiple choice type of questions can be asked always on this chapter for these all organic products, vitamins, enzymes, etc. Okay, now we'll talk about the microbes in sewage treatment, how they are used. It is, what is sewage? Everybody knows what sewage means, but still I'll just read out some of the part. It is a municipal waste containing human excreta, household waste, dissolved organic matter, and even pathogenic microbes like bacteria, viruses, protozoans, nematodes, fungi, etc. It also includes discharged water from hospital waste, slaughterhouses, slaughterhouse waste, animal dung, etc. Discharge from industrial waste contains toxic, dissolved organic and inorganic chemicals. Tannery, pharmaceutical waste, etc. also add to the sewage. See how much it includes. Sewage means what? What it includes? All the municipal waste that contains human excreta, household waste, then all other which contains household waste, contains so many other products also. Then it also includes the hospital waste, slaughterhouse waste, animal dung. Then also it contains discharge from the different types of industries. That means that is considered to be industrial waste, which contains generally toxic, dissolved organic and inorganic chemicals. Along with that, tannery, pharmaceutical waste, etc. That all contain sewage. So what the composition, you should know what that sewage is, what that sewage contains. All these industries means not only household, but hospital waste, industrial waste, pharmaceutical waste, from the tannery, etc. All this along with including animal dung and house waste, etc. Then what is the composition? Sewage contains approximately 99.5 to 99.9% water and 0.1 to 0.5% inorganic and organic matter in suspended and soluble form. Composition of sewage varies depending upon the type of waste discharged into water from different industries. For example, there are different industries like textile industry, chemical industry, pharmaceutical industry, dairy industry, canning, brewing, meat, packing, tannery, oil refineries, and meat industries. There are different types of industries, right? So according to the type of waste released from all those industries, these 0.1 to 0.5%, whatever the suspended matter is there, or that or soluble form is there, that it differs from this because of this all different types of industrial wastes. So according to the type of waste discharged into the water, these differs. The microorganisms which are present in the sewage, they are playing very important role. Various types of microorganisms are also present in sewage. Bacteria, viruses, fungi, protozoa, nematodes, algae are found in sewage. However, their number and type fluctuate depending on the sewage composition and source of sewage. Like the composition of sewage depends on the different types of industries, the microbes also, they differ as per the type of sewage. Uh, which type of sewage is there, from which type of industry that sewage is there, accordingly microbes also differ. So source of sewage is dependent, source of microorganisms or the microorganisms found in the sewage depend on the type of sewage or the composition of the sewage it contains. So raw sewage may contain millions of bacteria per ml. These include coliforms, fecal streptococci, anaerobic spore-forming bacilli, and other types of originating in the intestinal tract of humans. Before wastewater is made available for human use, it has to be treated properly so as to remove organic matter, inorganic salts, and pathogens as well. So now what are the basic steps involved in the sewage treatment we will discuss. Now we discussed about what sewage means, what it contains, what is the composition, and what are the microbial sources, right? Depending on the type of sewage, the microbes also differ. Now how this sewage water, which water is almost 99%. So what it contains according to the type of sewage and the industrial or the type of waste it contains, what treatment is to be done, which are the common steps used in the sewage treatment. There are basically 3 to 4 steps. We'll discuss one by one. Now preliminary treatment is what? Preliminary treatment, it includes screening and grit chamber. Okay, what does screening means and what is that grit chamber we will see now. Sewage and wastewater contains plenty of suspended, floating materials, pores, and solid particles along with dissolved substances. The suspended objects are filtered and removed. This is done in screening chamber. The sewage is passed through screens or net in the chambers. Larger suspended or floating objects are held back in the screening chambers. These have not removed, have to be removed before the biological treatment. So preliminary treatment includes the first step as screening, where large sized particles or the objects or the suspended particles, they are filtered and removed. So first step is what? Filtration in the screening. Okay, they are filtered. Large size particles are easily filtered using different types of nets. Different size of the nets are of different sizes are used, and these all objects, these all suspended particles, they are removed. They are filtered and removed. So filtrate is used, passed forward, so that sewage is passed through screens or net in the chambers. Larger suspended and floating objects are held back, they are removed. Okay, they have to be removed before biological treatment. In the just a minute. The next is what? Grit chamber. After screening, the filtered sewage is then passed into series of grit chambers, and these chambers contain large stones, that is pebbles, and brick ballasts. Coarse particles settle down by gravity. Thus, passage of filtered sewage removes much of the coarse particulate matter. Now, what happens then after filtration? That filtrate is taken to the grit chamber. Okay, that filtered sewage is passed to the series of grit chambers where these large size stones are present. They are called as pebbles, right? And then brick particles are present. Okay, so coarse particles settle down. With higher the gravity, they settle down, and thus filtered, which is remain on the top side, that is the as per the gravity, the large size particles settle down, and the passage of filtered sewage removes much of the coarse particulate matter. So large size particles are easily caught, they settle down. So after this preliminary treatment, the main treatment, that is called as primary treatment. Preliminary treatment includes screening and gritting. That means screening, that is what? Filtration is done. Then settling down, they are also called as settled chambers. So grit chambers. So large size or these heavy particles, they settle down. And now the actual treatment, that is the primary treatment, that is called as physical treatment. After the preliminary treatment, the sewage water is pumped into the primary sedimentation tank. Sedimentation of suspended solid or organic matter occurs in this tank. About 50 to 70% of the solids settle down. There is reduction of about 30 to 40% of the coliform organisms. The organic matter which is settled down is called primary sludge, which is removed by mechanically operated devices. The supernatant, that is the effluent in the primary sedimentation tank, still contains large amount of dissolved organic matter and microorganisms, which can then be removed by secondary treatment. So after filtration and grit or gritting or the grit chamber, when the sewage comes to the primary treatment, which is called as physical treatment, where large sedimentation tanks are there. That is, these are called as primary sedimentation tanks, where in these sedimentation tanks, the sedimentation of the suspended solid or organic matter occurs in this tank, and 50 to 70% of the solid settle down. Whatever the solid part is remained, again after gritting, after screening and gritting, after the preliminary treatment, again in these sedimentation tanks, maximum almost 70%, 50 to 70% solid gets settled down in the sedimentation tank, and that is called as primary sludge. Whereas the upper part of that means the filtered part or the supernatant, you can say supernatant, that is called as effluent. So in the primary sedimentation tank, still that contains the lower part which is settled down called as primary sludge, and the supernatant part that is called as effluent, that contains large amount. So what effluent contains? It contains organic matter and some microbes. And now this organic matter, that is the effluent, that is a supernatant containing microorganisms and organic matter, is passed to the next tank, that is for the secondary treatment. So what the secondary treatment? That is called as biological treatment. First primary treatment is physical treatment. Secondary treatment is biological treatment. In primary treatment, what is done? That is done with the help of sedimentation tank. It is allowed to settle down. So it shows two parts. The lower part, which contains, that is called as primary sludge. All the solid material gets settled down, and the upper part, that is the supernatant, that contains all water, including the organic matter and the microbes. Now this effluent, which is the supernatant, is passed for the next secondary treatment, that is called as biological treatment. So primary effluent is passed to the large aeration tank, and it is constantly agitated mechanically, and air is pumped into it, and aerobic bacteria grow vigorously and form flocs. Flocs are the masses of bacteria held together by slime and fungal hyphae to form mesh-like masses. These aerobic microbes consume the major part of the organic matter present in the effluent as they grow. And due to this, BOD, that is biochemical oxygen demand of the effluent, is significantly reduced. So physical treatment is done in sedimentation tank, and next to that, whatever the supernatant called as effluent is produced or remains, that is transferred to the secondary treatment to the large tanks that for the biological treatment. So this primary effluent is passed into large aeration tanks, where these aeration tanks, continuously aeration is made available. That means what? Agitation and aeration, both the things are done in this tank for biological treatment. Agitation, that means what? Airflow is started in the tank. That means air is forcefully pumped into the tank, as well as these tanks are continuously shaken vigorously. They are in the agitated. That means continuously, vigorously they are agitated as well as aerated. So aerobic bacteria, they work over there, and all these aerobic bacteria, they grow vigorously, and the flocks are formed. That means these are nothing but the masses of bacteria. Flocks are nothing but the masses of bacteria held together by slime and fungal hyphae. That means whatever the fungal hyphae are present, these bacteria are caught into those fungal hyphae and form a mesh-like structure. That means network, very fine, minute mesh-like structure, like masses they are produced. And because of this, what happens? Maximum organic matter, whatever is present as a waste, that gets stuck into caught into that particular those meshes, okay, these masses. And this aerobic microbes, because of these microbes, they consume maximum major part of the organic matter, and because of that, what happens? BOD, that is biological oxygen demand of the effluent, is significantly reduced. Now, after the biological treatment, secondary treatment, the last treatment, that is called as tertiary treatment. Once the BOD of the waste is reduced, it is passed into settling tank. And here the bacterial flocks are allowed to sediment. Whatever the meshes, that means these fungal hyphae, including these microbial masses, which are caught into the fungal hyphae, they are settled down in the tertiary treatment, because of their growth, BOD is reduced. And when the BOD is reduced, obviously it is passed to the next treatment, that is the last tertiary treatment, where they are called as settling tank. Here bacterial flocks are allowed to sediment. Sediment is now called activated sludge. Small part of this is passed back to the aeration tank, and the major part is pumped into large tanks called anaerobic sludge digester. In these tanks, anaerobic bacteria grow and digest the bacteria and fungi in the sludge, and during this anaerobic digestion, gases such as methane, hydrogen, sulfide, CO2, etc. are produced, and effluents from these plants, that is digester, after chlorination are released in natural water bodies like rivers, streams, and chlorination kills pathogenic bacteria, and digested sludge is then disposed. I'll repeat again the tertiary treatment. So after the secondary treatment, that effluent with the reduced BOD, that is biological oxygen demand, that is transported or that is passed into the settling tank for the tertiary treatment. Now what happens here? These flocks, that means bacterial flocks, they are allowed to sediment, and this sediment now is called as activated sludge. And this activated sludge, after the anaerobic means after the action of anaerobic bacteria over there, what happens? This anaerobic sludge, it gets digested, and it remains at the bottom, and afterwards it is removed and used as a fertilizer. Whereas the effluent which is present, that can be released in the natural bodies like rivers and streams, and during that anaerobic digestion, where anaerobic bacteria, when they are working for the tertiary treatment, these different types of gases are released like methane, hydrogen sulfide, carbon dioxide, etc. They are released, they are produced during this anaerobic digestion. And effluent, that means the supernatant, which is that nothing but the water that contains whatever that water remains, that is called as effluent, that is then released to the natural water bodies like rivers and streams. And after that, that if water is to be used, that can be chlorinated. So chlorination kills pathogenic bacteria, and the digested sludge which is produced, that can be disposed of, and that can be worked as a manure or the fertilizer in the farm. So these are the treatments. I will not continue because this is very important thing which is shown here in your textbook also. Sewage treatment, screening, grit, this is what preliminary treatment which removes the heavy solids. Then primary treatment, removal of the suspended solid particles. Then secondary treatment where in the secondary treatment, that is biological treatment, primary is physical, secondary is biological. Aeration and agitation, both the things are done. Aerobic bacteria work over here so that it gets converted into activated sludge, and for the that is the sludge part. And the effluent, that is a supernatant, that is treated and that can be recycled, and that activated sludge, by the action of anaerobic bacteria, it gets digested. Anaerobic bacteria work over there in the tertiary treatment, and then that sludge which is produced, that is used for as a manure and or that sludge cake to dispose. That means it can be used as a manure or biofertilizer. And this, by after the tertiary treatment, whatever the effluent is remains, that water is recycled, it is chlorinated, chlorination is done to kill the pathogens, and then can be released in the natural water bodies. So this is what very simple method you can remember in case of sewage treatment. All the three steps are used. These are some of the just pictures to show how this it contains municipal waste called as sewage. These are the sewage treatment plants. So primary treatment in small scale, it is shown. Some of the pictures for this is what the grit, that is removed by sedimentation, that means soil and small pebbles. And after that, primary treatment, this is what sewage, this is the sludge, and this is the effluent, this is the suspended, and this is the sedimentation, suspension and sedimentation. And these are some of the diagrams which shows the different manufacturing means which are used for the treatment. These are some of the diagrams or the pictures which show different that material or the plant, in the plant whatever is used. So these are the masses of bacteria, it is shown. DOD is nothing but the amount of oxygen that would be consumed if all organic matter in 1 liter of the water were oxidized by bacteria. So this is what just what BOD, how it is useful to reduce the BOD is very important. And this last part, activated sludge, that can be removed, and the final sedimentation output. So in the small scale, it is shown anaerobic activated sludge. So this is what all about this sewage treatment, and I will stop my sharing right now so that if you have any doubt, you can ask me